Hi everyone, my name is Norhaizad. I'm from Bachelor Science Biotechnology with Anna. I will do a Pecha Kucha for microbiology subject. So I'll be talking about a fascinating organism which is Saccharomyces cerevisiae. They're also known as brewers or vacancies. This tiny organism has significant impact on our daily life and scientific research. So Saccharomyces cerevisiae is a species of yeast that belongs to the kingdom fungi, phylum Ascomycota, class Saccharomyces, order Saccharomyces, family Saccharomycetaceae, and genus Saccharomyces. Saccharomyces cerevisiae is a species of yeast. Yeast are a single-cell fungus microorganism. Saccharomyces cerevisiae cells are around to avoid 5 to 10 micron in diameter. They are microscopic, which means you need a microscope to see them. They are unicellular eukaryote that reproduce by a process called budding. They are flat, smooth, moist, glistening, or dull, and creamy color. So, this species has been instrumental in wine making, baking, and brewing since ancient times. It's been used for thousands of years to ferment sugars, producing alcohol and carbon dioxide. It is believed to have been originally isolated from the skin of grapes. This is why it is so important in winemaking. Saccharomyces cerevisiae is one of the most intensively studied eukaryotic model organisms in molecular and cell biology. This means that it's used as a representative organism to study how cells work. So, there are so many proteins important in human biology. We were first discovered by studying the homologue in yeast. This includes proteins involved in DNA repair and cell cycle control, and also protein synthesis. Saccharomyces cerevisiae is currently the only yeast cell known to have Berkeley bodies present, which are involved in particular secretory pathways. These structures help the cells secrete proteins and other molecules. So, the medical relevance for these microbes, antibodies against Saccharomyces cerevisiae are found in 60 to 70% of patients with Crohn's disease and 10 to 15% of patients with ulcerative colitis. This suggests that this yeast may play a role in this disease. So, Saccharomyces derives from Latinized Greek, means sugar, malt or sugar fungus, while Cerevisiae comes from Latin and means of beer. Other names for organisms include brewer's yeast, ale yeast, top fermenting yeast, baker's yeast, roguite yeast, and budding yeast. This name reflects various means. This species is also the main source of nutritional yeast and yeast extract. Nutritional yeast is a popular food product used for its cheesy flavor and high protein content. So, Saccharomyces cerevisiae in the 19th century, great bakers obtained their yeast from beer brewers. This was before the development of commercial yeast production. So, Vienna process was developed in 1846, which included better processes for growing and harvesting top fermenting yeast. This improved the consistency and quality of yeast. So, Saccharomyces cerevisiae is facultative anaerobe that metabolizes glucose through fermentation or respiration. It can also metabolize other sugars such as fructose and galactose. It is also trophic for certain amino acids, meaning it requires them in its diet. So, Saccharomyces cerevisiae contributes to alcohol and baked good productions by undergoing a biochemical process known as fermentation. In this process, it converts sugar into alcohol and carbon dioxide. So, what's the natural habitat for these microbes? Saccharomyces cerevisiae naturally grows on fruits such as grapes and dates, as well as grains like wheat and barley. It's often found whenever sugar is available. So, the impact and what the application from this. Saccharomyces cerevisiae has been used in winemaking, baking, and brewing since ancient times. It's also used in the production of biofuels such as ethanol and butanol. It is a model organism in genetic research. It is also used as a dietary supplement and has potential health benefits. So how it's produced? Its primary form of reproduction is budding, where daughter cells sprout directly off of mother cells. This is different from how many cells divide, which is by splitting into two. So, since it is have been used in the ancient, some of the oldest known use of Saccharomyces cerevisiae goes back to 4000 BCE in ancient Egypt, where ancient Egyptians would use yeast to make a bread. This is one of the earliest forms of biotechnology. So what's the benefit of the biological studies? As a rapidly reproducing eukaryote, it is widely used for modern organism that has allowed scientists to better understand molecule, cellular, and biochemical process. Its genome was one of the first to be fully sequenced. So the SDG contribution is it contributes to several of United Nations Sustainable Development Goals. For example, it is used in the production of biofuels, which can help reduce greenhouse gas emissions and mitigate climate change, which is SDG 13. So in the conclusion, Saccharomyces cerevisiae, though tiny, play a huge role in our daily lives and scientific research. From the bread we eat to the wine we drink, and even the medicines we take, these humbly made significant contribution. Thank you for your attention.